Welcome to this pre-class video on membrane lipids. What are membrane lipids? Well, let me draw you a kind of a cartoon of a membrane lipid and highlight the components of a membrane lipid. And you definitely need to know these components. So it starts with glycerol, and we have seen glycerol previously when we talked about tags. And then attached to the glycerol backbone, remember glycerol is a triol, has 3 hydroxyl groups. Attached to that, or esterified to that actually, are 2 fatty acids. And again, this is my kind of a cartoon for a fatty acid attached to glycerol. Here's another one. And these fatty acids can be saturated or unsaturated. There we go. Also attached to the glycerol is a phosphate group and another type of alcohol. And so these are the components of a membrane lipid: glycerol, 2 fatty acids, phosphate, and an alcohol. Sometimes we're probably going to draw this even more shorthand like this. Kind of a circle with 2 squiggly lines on it. Well, what is this? This is what we call the polar head group. And it really represents the alcohol plus the phosphate. That's what makes that this part of the molecule polar. And then these 2 squiggly lines really represent the non-polar tails. That's not one word, so let me put a space there. Non-polar tails of the fatty acids. So this is kind of the architecture, if you will, of a membrane lipid. A polar head group, 2 non-polar tails. Well, these things like to what we call self-assemble into a bi-layer, 2 layers. And we see these in, for example, in a cell wall. If you've had a biology class that talked about cells, you've talked about the cell wall. What is the cell wall? Well, you need to keep things out of the cell, and you need to regulate what can come in and go out of a cell. A bilayer is a good way to do that. So let's talk a little bit about how do you make a bilayer. Well, let me kind of draw a cartoon, and you can see that the non-polar tails of individual membrane lipids like to come together, and they can make a bilayer. So here we go. So here's a membrane lipid, and I'm going to keep drawing them. So that's one layer, if you will. Another layer comes along. Now let me draw this in kind of the gray maybe, although you can't really tell the difference, so that seems to be doesn't matter. Let me just draw the other layer of the bilayer. Again, I'm just adding membrane lipid after membrane lipid. And if you kind of, it can certainly go on and on and on. What we have here is certainly a bilayer. We have 2 layers. We can split it right there. If this is inside the cell and this is outside the cell, then this is what we call the outer leaflet. That's the terminology. Learn the words here. Outer leaflet. This is called the inner leaflet. And so there are definitely 2 layers here. The outer leaflet and the inner leaflet, and they come together at this interface, and they are held together by what we've called before dispersion forces. And this whole area right there, right in this right there is highly nonpolar. So it does not allow polar molecules and ions, things that have charge, to freely cross the membrane, the bilayer. Things that are more nonpolar are more likely to cross by itself. But proteins and ions, let me go back. Polar molecules plus ions, they need a transport protein. And these transport proteins are actually embedded in the bilayer. And so these polar molecules and things that have charge, we call them ions, they're just not going to be interested in interacting with the nonpolar part of the bilayer. But if there is a protein molecule kind of embedded in the bilayer, then polar molecules and ions can go through without touching the nonpolar part of the bilayer. And these transport proteins play a very important role in cellular biochemistry. Again, this is kind of a pre-class video, and we'll get into a little bit more detail in class. I hope this video was helpful.